Good morning, my dear students. Today I'm going to talk about periodic structure and the alpha helix. This is my second week and it is the eighth lecture. Myself, Mehdi Hayas Shahi, Assistant Professor at Interdisciplinary Research Center. This is in Faculty of Medicine, Aligarh Muslim University, Aligarh, Uttar Pradesh, India. In this lecture, I'm talking about geometry of alpha helix, then how it gained the stability and its variant, and also the biological roles of the alpha helix in the system. The content of my talk today is alpha helix discovery, and it was Pauling and Corey in 1951 X-ray crystallography with the help of X-ray crystallography. Then I'm going to talk about hydrogen binding pattern, how this alpha helix has hydrogen binding pattern, and normally it has I arrow I plus 4 rule. What it does mean the rule? I represent any amino acids residue in the polypeptide chain. The number after the arrow is like I plus 3 or I plus 4, I 5 plus tells that how many residue ahead of the hydrogen bond is formed. I3 means 3 residue ahead, I plus 4 means 4 residue ahead, and I plus 5 is 5 residue ahead. Then I'm going to talk about side chain orientation. Outward projection, the periodicity is almost 3.6 residue. And then we discuss about helix stability factors. What are the factors which really give helix stability? This is a capping, salt bridges, and helix breakers. Then going to talk about geometry and parameters like rise of helix, then pitch, residue, turn, bonds, length. Then I'm going to discuss about Ramachandran plot or space, talk about phi and psi values, then conformation restrictions. Then I'm talking about different variant of helix, that's 310, then we have pi helix and polyproline and collagen. Then also discuss biological function because like DNA binding and also membrane protein and also motor proteins involved in this. References I concerned for this lecture, biochemistry, note 8th edition of Liberty Triad, and also Harper Illustrated Biochemistry and Principle of Biochemistry by Albert L. Lellinger. Now it turns helix types, and there's a three main helix type: alpha helix, then 3-dentate helix, and pi helix. These three are different helix. Alpha helix hydrogen bond pattern I to I plus 4 means fourth residue, and it means carbonyl oxygen CO of residue I bonds with amide hydrogen NH of residue I4. And then 310 helix, it is I plus 3. It means H hydrogen bond between residue I and I plus 3. Then pi helix, it is I5, I plus 5. It means hydrogen bonds between residue I and I plus 5. It is the characteristics of alpha helix is most common. Whenever the alpha helix, so this is most of the common one. And that's 3.6 residues per turn. 310 helix is a very tighter and also narrow helix, narrow in nature. Then pi helix is wider and loose helix is a loose helix because I5 is binding at fifth hydrogen position, binding hydrogen position in bond. Timeline for alpha helix discovery. In 1930 to 1940s, almost 10 years, Asbury coins alpha keratin from X-ray fiber pattern of keratin. They got alpha keratin pattern, and in 1948 to 1950, Pauling folds a paper strip and envisions the helical backbone. They is given the concept of helical backbone. In 1951, Pauling and Corey published alpha helix model, and it was published in Proceeding of National Academy of Science. Is very reputed journal. Then in 1950, Max Percius confirmed the presence of alpha helix in myoglobin with the help of X-ray crystallography. Use the X-ray crystallography. What is the key contribution by Linus Pauling and Max Percius? They adapted non-integer periodicity, not 3 residue per ton, not 4. They adjust exact value. They have given 3.6 residue per ton, and they avoided any force integral symmetry. Then they built accurate physical model because they have very accuracy because of exocrystallography using precise bond length and also angle derived from small molecule structure. That's very much clear. This is Riemann depiction of alpha helix and this cylindrical depiction of alpha helix. And the confirmation they got it from X-ray crystallography. So Max Burgess added Pauling's 1.5 angstrom reflection as a X-ray pattern of horsehair. In this person confined the presence of alpha helix in real protein, means alpha helix in the most of the protein they presented there. The helix was identified in the first solved structure, it is myoglobin, in 1958 by Kentrew. Then in 1960 by purchased in the hemoglobin. So first is myoglobin, then hemoglobin. They resolve the structure, protein of helix sulfide structure. This is the helix direction. Most of the helix is right directions, means right-handed clockwise directions, and also very few are left-handed directions. Right-handed helix are more stable and has less steric hindrance. That's why most of the helix are right-handed helix, and almost all natural proteins helices are right-handed helices. This is one example is here ferritin, structure of ferritin. You can see this is a right-handed helix. This is the turned helix of alpha helix shows this showing here. Now geometry and structural parameters. So it is has 3.6 residue per turn, mean that non-integer periodicity characteristic feature of the alpha helix. See, this is 3.6 means 1, 2, 3 and 6. The 3.6 residue, not complete 4 or not complete 3. The 3.6 residue per turn, and each rise means 1.5 angstrom of this each rise is a translation along the helix axis per amino acids. Then it is 5.4 angstrom pitch, means each turn as a distance is 5.4 angstrom. That's per turn is a distance along the axis of one complete turn, that is 5.4 angstrom, and it has 3.6 residues. Then also the rotation per residue is 100 degree, means that is the angular rotations of the helix axis for each amino acid is 100-degree rotations, and the diameter it is 2.3 nanometer of the helix diameters, overall width of helical cylindrical. Now the translational rise, what is translational rise is denoted with D. Each carbon alpha atom advances 1.5 angstrom along the helix axis per residue, means per residue is 1.5 angstrom. For a typical helix of 10 residue, overall length is 1.5 nanometer. That is 1.9 nanometer. Now it is a rotational angle. Rotational angle is adjacent C alpha atom rotate for 100 degree about helix axis. Suppose helix axis is completely a 360 divided by 3.6 residue, it's become 100 degree. Ensure that H bond donors and acceptors aligned across 4 residue, aligned 4 residue. And now the pitch, one full length means one full length is 360 degree turn of helix, and it has span 3.6 multiplied by 1.5 angstrom. It is equal to 5.4 nanometer or 5.4 angstrom or 0.54 nanometer. So this is the one pitch and this is the C axis. C axis. Repeat identified by X-ray crystallography diffraction as 5.4 is a reflection, means repetition of the helix. Now the L-helix, which I already explained in my previous lecture, most of the amino acids are L-amino acid, only right-handed helix is favored. For L-amino acid, right-handed helix is favorable because of less sterically inhibitions. And the left-handed alpha helix is theoretically allowed but exceedingly rare. Means left-handed helix is not possible. Some point is possible only in the presence of glycine amino acids in the helix. Now the hydrogen bonding bond patterns, what's the donor and receptor? So donor, the amide NH residue of I10, NH residue of I10 donates the hydrogen bond, donates the hydrogen, and this NH bond is directed toward the acceptor carbonyl. An acceptor carbonyl CO of residue I accept the hydrogen bond and the oxygen lone pair is the electron donor in this case. And what's the geometry? NO distance. NO distance becomes 2.8 angstrom and NHO angle is almost 155 to 160 degree. And these are the near ideal H bond parameters. This is the hydrogen bond. You can see here Ri is here. Then they have a binding is here, hydrogen bond. So from here to here, hydrogen bonding. So this is ideal pair. And the strength of this hydrogen bonding, because it provides the strength, and each backbone hydrogen bond contribute 8 to 20 kilojoule per mole. And a 10 residue helix forms 6 hydrogen bonds. And this substantial, and this is because of 6 hydrogen bonds, this is substantial strength the helix, alpha helix, cumulative stability. And also what the periodicity of this condition of alpha helix, because each turn is span 3.6 residue and each hydrogen bond span 4, there are 3.6 divided by 1 is equal to 3.6 hydrogen bond per turn per residue, means 3.6 hydrogen bond per turn and one hydrogen bond per residue. And all hydrogen bonds run in parallel to the helix axis, means they are parallel in nature. And this alignment maximize their electrostatic component and create a helix as a macro dipole, just like a water in dipole there. It also created a macro dipole that is also giving the high stability and also help in dissolving the water. Now alpha helix in Ramachandran space. I already explained the Ramachandran plot that is going to define the rotation of the peptide, means alpha helix or any proteins, and they also give the restriction and they favor only limited, only 25% of the conformations. Which I previously explained. This is bi-axis has the psi and then phi is x-axis. Minus 80 is the maximum and plus 80 is the also, and this is the minus 80 is the minimum and plus 80 is the maximum angle rotation, minus 80 y-axis and plus 80 y-axis. So what happened, right-handed helix is very common in the range of minus 60 of psi and minus 60 of phi. This is a range that for right-handed helix, and left-handed, and this is very common. And the left-handed helix is very rare. It's very rare, and it is in it is in the range of plus 60 and also plus 60 of phi and psi angle. That is very rare. Alpha helix, the most preferred reasons is minus 57 degree phi and psi minus 40, and is upper left quadrant lower hemisphere. This is the right-handed helix. Favored conditions. Ideal torsion angles. What is the ideal? Phi in the range of minus 40 to minus 70 degree, that is a phi. And the rotation of alpha carbon and N bond is 57 degree. This is the ideal torsion angle, and that is fine. What about psi angle? It is -35 to -55 degree, and rotation of C alpha carbon to carbon bond at 47 degree. This is ideal conditions. And omega should be 80 degree, 180 degree, 180 degree, because it's a trans in nature, and also peptide bond is fixed. And also there is a deviation in the range of 10 to 15 degree from the ideal, and it is still maintain the hydrogen bondings. There is no issue with, and that's very regular bonding with hydrogen bond. There's no issue. 10 to 15 degree of deviations from Helix. Not the side chain on amphipathetic helix. The role of side chains and amphipathetic helix. Side chain are projected outside, just like here the side chain. This is a side chain. This is a side chain. A side chain projection is a green color. All side chain project readily outward and slightly downward. To just say, most of them, the side chain protrudes outward and slightly downward, mainly toward N terminus from the helical axis. And because this reason, this projection, because of the, it minimize the steric hindrance between adjacent residue and also expose the interaction site. That's very important. And they also follow 100-degree rotation rule, because adjacent residue rotate 100-degree about the axis. The residue I and I plus 3 plus 4, I plus 7 cluster on one face. And in amphipathetic helix, hydrophobic residue cluster on 100-degree face, while hydrophilic residue cluster on one another. This is a characteristic of 100-degree rotations. Protein at lipid water interface, when the protein interact with lipid water interface, is expolite the amphipathetic helices. How it does? The hydrophobic phase insert into lipid or membrane surface, because of a lipid is also hydrophobic, while the hydrophilic phase remain in aqueous solution. And example, apolipoprotein, antimicrobial peptides, manganine and melitine. These are the protein amphipathetic helix. And this is a coiled and coil heptide repeat. What it does exactly? In this case, in coiled and coils, example is tropomycin GCN4 leucine zipper A7 reside heptide repeat like B, A, C, D, E, F, G, placed hydrophobic residue at A position in the front, and the D position at one face, and driving interhelical packing. This is a direct sequence of 3.6 residue per turn geometry. This is unique geometry of coiled and coil heptide repeat. Now the helix variants. What are the helix variant? There is a alpha helix, then 310 helix, then pi helix, and also there is a polypro second helix. The pattern of H bond pattern in is I and I plus 4, because it's going to bind in the fourth residue of hydrogen bond. But in 310 is I plus I3 in the third residue, and pi helix I arrow I plus 5 in the fifth residue. But polypro second has no hydrogen bond. In this case, there is no hydrogen bond. Then residue per turn is 3.6 in the case of alpha helix. Then 310 has three residues per turn, and pi helix has 4.4 residue per turn, and polypro second has 3 residue per turn. And rise, the residue, means how much the length of that each turn this become 1.5 angstrom for alpha helix, then 2 is the 310 helix, and 1.5 pi helix, and 3.1 is angstrom is for polypro helix. Now the pitch, the complete turn, how much distance covered? 5.4 in the case of alpha helix, and 310 is 6 angstrom, and then 5.1 angstrom in pi helix, and 9.3 is very angstrom in polypro. This is very lengthy one. The ideal rotation, torsion angles, rotation for phi and psi, -57 to -47 degree for -47 is for phi and -47 psi for alpha helix, -49 and phi and -26 psi for 310 helix, then pi helix -57 and -70 degree psi angle, then -75 pi and 150 psi angle for polypro. And what does sense? Alpha helix is right-handed, 310 helix is also right-handed, and pi helix is also right-handed, but polypro is left-handed. That is very rare, left-handed. Frequency is very common. 310 helix is rare, is 3.5% residues, and pi helix is very rare, that is very rare, and polypro mainly in the collagen, and it is very disordered in nature. And diameter 1.1 is alpha helix, 0.9 is 310 helix, and 1.4 is pi helix, and polypro is 0.6 nanometers. So what is sense that alpha helix is very common in this case, and pi is very rare ideally, and 310 is hardly 3.4%, and polypro is in collagen disorder, etc. Now, helix stability means how helix gain the stability. It is these are formers and also the breakers. Then helix capping, it has N cap and C cap. What is this N cap and C cap? N cap residue is in position N1. N1. The side chain donates H bond to the unsatisfied NH of residue N2 and N3, like in case of asparaginine, aspartic acid, serine, threonine are favored. In this case, in case of N cap, and the C cap, side chain acceptors like H bond form with CO and of C3. Threonine, asparaginine are common, and capping can stabilize helix by 2 to 8 kilojoule per mole. This is also giving a stability to the helix. And glycine is a flexible disruptor, means it's going to be weak the alpha helix. Why it is? Glycine has no side chain. We all know that glycine has no side chain, and because of that is unusual conformation freedom of pi and psi angles quadrant, and this flexibility destabilizes the helix by increasing the entropy of the unfolded state. Means it's going to increase the entropy. If the glycine is there, it's going to increase the entropy because of unfolding the state. And how this helix is going to be salt bridge and I10 interactions. Glutamic acid I and lysine is I4. Aspartic acid and I and asparagine I4. It is intrahelical. It builds an intrahelical salt bridge and align along the helix surface and can stabilize helix by further 4 to 10 kilojoule per mole. It's further going to give the stability because of salt bridge with the help of glutamic and lysine, asparagine and arginine. Especially in the low dielectric environment. It helps in the low dielectric environment. Now, helix stability, formers and breakers. Why alanine is the best former? Alanine is the best former to give the strength of alpha helix. Alanine methyl side chain is small enough to avoid backbone class because single methyl group, and that's avoid the backbone class and provide mild hydrophobic stabilization. So, means providing also mild hydrophobic nature of the methyl group, and this C beta subtly stabilize the I4 hydrogen bond geometry because of these reasons. The beta sheet in this case is established as. And proline is the helix breaker, and the proline is helix breaker because proline lacks backbone of NH, no hydrogen bond donor, and its parolidine ring fix at minus 60. The angle pi, pi angle is minus 60 while imposing a rigid kink and terminates helices and introduce bend because of proline. Exception is occur in N terminus of helix where N cap and H bonds is treated. That is exception when the N and H cap bond is treated. Other example, beta carbon branch residue like valine, isoleucine and thurine. These are beta carbon branch residue. This strict hindrances destabilize the helix because they have a strict hindrance and they destabilize the helix. And serine, aspartic acid, asparagine side chain form competing hydrogen bond with backbone of NH and CO group. So they are giving more competing hydrogen bond with NH and CO groups. Now the biological function of helix, because the important why alpha helix is the characteristic because the biological function. It is this kind of helix is in the transcription factor and it's called DNA binding transcription factor dot helix turned helix. What it does, this motif HTH helix turned helix insert in the major groove of the DNA and a specific amino acid like interactions provide a kind of sequence recognitions and it is present in the more than 1000 transcription factors like RAP repressor, LAC repressor, Crow homeodomain transcription factor and other transcription factor, more than 1000 transcription factor which is going to bind with the major groups to identify the sequence. It also help in molecular motors and muscles like myosin S1 has domain which contain converter helix that amplify a small active site changes into large lever arm swings, generating 5 nanometer movement per ATP. And also tropomyosin regulate actin via its long coiled and coil helix. This is the characteristics of tropomyosin. Then there is a coiled and coil and structure rules. Two or more alpha helix wind around each other in left-handed supercoil driven by hydrophobic peptide repeats and provide mechanical strength in a structural protein because of this two helix coiled and coil and mediate protein-protein interaction in different signaling pathway or complexes. Now the membrane, another function of biological alpha helixes, membrane spanning helixes. Like hydrophobic alpha helixes has 20 residue and it is spanned through lipid bilayer and the length of 13 angstrom hydrophobic core. And this helix backbone satisfy hydrogen bond requirement in a low dielectric environment. Like GPCR have 7 transmembrane helixes and ion channel use helical bundle for the pore. All ion channels have helical bundle for the transportation of the ions there. Then also enzyme active sites. Helix dipole activates substrate bindings at the N-terminal end of helix like P-loop, phosphate binding loop, places the N-terminus of the helix over phosphate of ATP and JTP. Serine proteases use the helix dipole to activate the catalytic serine. Now and also it has antimicrobial peptides characteristics. Like cationic amphipathic helix like manganine, melitine and defensine selectively disturb bacterial membrane. The positive charge attract anionic bacterial lipids and the hydrophobic phase insert to form a pores or carpet model distributions of the bacterial cell wall. Now the summary of my my presentation or talk today, my lecture, alpha helix at gallons. Discovery, the Pauling and Corey in 1951 using model building and precise bond pattern parameter to identify and confirm by Persian X-ray diffraction with the help of X-ray crystallography diffractions. And there's H bonding, CO is I, then NHI plus 4, it is 2.86 angstrom, NO distance is 150 degree, and NHO angle is 150 degree, and then one hydrogen bond per residue. Dipole moment like 3.5 dipole per residue, then partial positive at N terminus and partial negative at C terminus exploited the ion and cation binding. Then also discuss about the stability control. Alanine is the best former. Alanine is the best former, and proline and glycine is the breaker. And then N and C, nitrogen and carbon cap residue, stabilizes the helix termini by satisfying the lone hydrogen bonds. Then the geometry is 3.6 residue per turn, and there's a 1.5 angstrom of the rise. Then 5.4 angstrom is the pitch complete helical turn. Then we have 100 degree rotation right-handed for L amino acid. Means all the most of the helix are right-handed with L amino acids. Then I discuss about Ramachandran plot or graph, like providing the rotations and the exact positions of different helix and also favorable conditions, like pi minus 57 centigrade and psi minus 47 degrees is the best suitable, lies in the upper left cluster of the Ramachandran plot of L residues for alpha helix. Then the helix variant discussed 310 helix, it has I and arrow I plus 3 hydrogen bonding. Then we have phi helix, it is I arrow I plus 5, then both rare and less stable than canonical alpha helix. Then discuss about biological role in DNA binding, helix turn helix loop. Then we have transmembrane helix for the transportation of ion channels and all that. Then we have a coiled and coil like in keratin and myosin for a strength to the muscles and protein there, a strength and very strong binding. Then also enzyme active site activation, because this has to and bind the receptors ligand to activate the means substrate and enzyme activators there. Thank you so much.